In this video, I'll talk about mass spec in biofield. Couple of my students intrigued by my comments on mass spec is also used for biomolecule, the big molecules. And honestly speaking, I cannot teach you properly for so many applications being done in the biofield, and the analysis of this big molecule is not as simple as small organic compounds, and you need a software to help you, in analyzing the signals and so on. But I can give you sort of a bridge video for you to bridge it over from organic level to bio level. So J.J. Thomson was the first person who did this experiment regarding charges, right? From Gen Chem, you heard about him. And since then, these physicists / chemists, they were interested in finding out the natural abundance of the isotopes, the relative amount. Then mass spec is used later in 1959 to sequence small peptide or small oligonucleotides. Then significant technological advance is made when we used Fab as ionization method. Then further we were able to do a better job when we had a hands-on electrospray ionization method around 1988 when I was graduating college, I guess. That was connected with the LC Mass when I was in the Michigan for masters. Yeah, many people were working on LC Mass. That was the one of the frontier method at the time. Then 1990s in Japan, non-PhD researchers in a company laboratory, he eventually got the Nobel Prize, but developed the MALDI method using laser. Then the MALDI is also connected with the time of flight analyzer, and that allows us to vaporize huge molecules like actual proteins. So nowadays we are able to get the molecular weight of the protein. Not only that, we can identify protein by checking their distributions or the signature peaks of certain amino acids and things like that. Then the further development is made that we by connecting mass spec with another mass spec, basically the analyzer with another analyzer, right? So you can do mass spec and you get a certain peaks, right? Bunch of peaks. And then they're so big numbers because they are big proteins, and after you fragmented them, they're still huge. But you want to look at this peak in particular. I think I explained it in other video, the method like ion trap or time of flight quadruple analyzers. You can have them travel around, okay, in a cup or in a tube, and keep them, and you can send them out one by one by the mass, and you can measure them out. But if you click on the computer to select this fragment, then the machine will let this fragment out of the trap, for example, and let it move to the next ion cup, ion trap, then have it get fragmented more there, then you can see whatever the sequence of this amino acid was, you can observe further fragmentation, and you may get smaller fragments. That's making this relatively small sequence of the amino acid or DNA or sugar molecules or whatever. Then you get all these molecular weights of the smaller segment of the peptide, for example, and then say you like to look at this one further, then you can have this ion trap send out this one as you select, and you can connect more. Then you can observe it as it fragmented more, and you can keep doing it until you get eventually single amino acid, and you can get exact amino acid molecular weight here. Such as these days high resolution mass spec can give you this kind of number for single amino acid. For example, glycine 57.02147, this accurate number. And other common amino acid like alanine is some of the small ones like this, and so on. You can find the table for it, so you can read these numbers here, but then they were here in the longer segment of the peptide, right? So say this was like 625 or point something something something, then this was originally from this segment. I don't know, it could be 4528 point whatever, and you can eventually combine all these data, and then getting the information about the molecular weight and or something like primary sequence of the protein, or whatever portion of the sequence has been changed or tagged or modified. You can apply whatever way you want. And this is the current status of the mass spec. So this tandem mass spec used to be like MS or MS square, but nowadays you can connect many more. And what makes the interpretation of the mass spec from the biomolecule so difficult is because you expect too many peaks, and also almost never you get the molecular peaks. And I mean, it depends, but the reason being, for protein, for example, as you know, some protein side chains are always positively or negatively charged in a certain pH. And then they change number of positives and negatives depends on how you prepare sample, what buffer you are using, what pH buffer you are using. So protein is never +1. It has multiple pluses already. So you need to use a certain mathematical methods to figure out what each peak here and here what amount of the plus charges you actually have for these fragments. So it could be end up you finding using certain algorithm of calculation, this 1100 peak was the one that has a 14 plus, and then this something around 900 was about 16 plus, and then this must be, it's like a 15 plus, and whatever, it keeps going on and on. So eventually you can finally deduce the mass when the charge was +1, although you don't see it. So this is when sample, the protein sample is put it in the machine. But sometimes, you know, you're using restrictive enzyme, digestive enzymes to cut a certain area and then isolate them, say in a gel, right? Something like that. Then you do blotting, for example, like for you know, DNA will do sudden blotting, and for proteins you will do Western blotting. Then for RNA you do Northern blotting. For Eastern or Easterners we do post-translational modification. Or, you know, RNA, DNA, lipid related, and all kinds. Then we get a certain sequence that we know, put it in the machine, get the mass back, and you can do tandem, and you get the data. Then you can compare. And some of the data you get with the known proteins, they are already in the database, so you can get the information, the database information, and you know, compare to figure out what you have or what changes been made, and you know, and so on. And here are the some of the researches adopting the mass spec in the biofield, and I have some reference. If you're interested, you can search them up. These are about like 10 years or more, like 15 years old reference. So you should know a lot of advanced been made. However, I hope this is useful for you. So some mechanistic study, you know, like a fatty acid metabolism, or you know, any kinds of mechanistic study, or when you study about the nutrients, how it is absorbed, and about the retention of it, or how it's utilized in the body. We put isotopes and then, you know, take the sample and from the body, and use the ratio change to come up with a mechanism. So this journal here will have the detailed information about it. And others is like a small organic molecule in vivo for a pharmacokinetic study. People use mass spec, and the data is here, Analytical Science 2003 journal. And the macromolecules like DNA and RNA and protein, you can get the information here, like DNA sequencing is done, and the data is published here in JACS 2003. Or when people study the fatty acid oxidation disorder for a newborn, how the instrument is used, the research is published here. Then about proteins, right? Identification, even in the femtomole range, using electrophoresis, right? So this is journal name, by the way. And also one of the main things happening these days when you study for such as phosphorylation, in the switchings and signalings of the cells, such as like cancer studies, right? Glycolysis mechanism. All these post-translational modification research, adopting mass spec. Actually, one of our faculties in the college, he's expert in the MALDI-TOF, and he's studying mechanism on the phosphorylation, but he's actually developing the methodology of this instrument to apply and calculate how much phosphorylation is actually taking place. And like I said, the data processing is not simple, so he's trying to improve the process, and that is also published at Account in Chemistry Research 2003, so you can look it up. So hopefully I gave you a little bit of a bridge knowledge from chem to the bio about the mass spec. And again, if you have any more requests, please email me. Thank you.